Good afternoon. You're here on Likeable Science on Think Tech Hawaii, broadcasting from the Pioneer Plaza studios. Likeable Science is all about the fact that science is a fascinating, fun part of everyone's life, and we all should relax and enjoy it, and help others see that science can be and is really not a foreign, scary thing, but just something to be explored, to be learned from, to be enjoyed. With me today to help explore this further is Dr. Mary Hagedorn from the Smithsonian Conservation Biology Institute. She works over on Coconut Island here on the other side of Hawaii, and does exciting work on coral preservation through conservation through cryopreservation.
It's quite a tongue twister.
Welcome, Mary.
Thank you. It's a pleasure to be here, Ethan.
Mary is a renowned scientist who travels the world doing all kinds of interesting stuff, collaborating with folks in Australia as well as in the Caribbean, setting up coral banks of coral germplasm of all sorts, has dived extensively, watching the changing sizes and populations of coral reefs in a number of areas, does very wide range of interesting things. She had us over to her lab some months ago, where we got to help do artificial insemination of coral, I guess you would call it, extracting coral sperm. So that was a fun evening. Mary got her doctorate from Scripps and has been now with the Smithsonian for about 20 years, I believe. And she teaches classes, gives talks around the world to all kinds of audiences, scientists, but lay audiences of various sorts, Star Trek people, all kinds of people. And she's an accomplished grant writer. She's won many awards for her work. And so let's just jump right on into this. And let me just start by asking, what got you started on coral? Why coral?
Well, it's actually more a personal reason I got into studying coral rather than a scientific one. I had been working in the Amazon for many, many years, working on fish, electric fish actually, and we had a very tragic expedition where the boat we were in sank and a number of people were killed actually. And so I wasn't on the boat right at the time when that happened, but I realized I couldn't go back to that kind of work, that line of work. And I liked being outside. I loved diving. My husband loves to dive, and I thought, coral reefs sound good. And I knew coral I had been working on trying to cryopreserve fish, and I knew coral were struggling, and that the threat to coral reefs was looming, and that we had to develop the science before the threats got to the point where we had no time left. So working on cryopreservation and developing the actual science to do it has taken a number of years, and so that's what I've been doing.
Very interesting. So that sort of leads then to the next question, sort of, you know, why coral? I mean, who cares about a bunch of rocks, right?
Well, the first thing everyone in the audience, I hope, will learn and take home is that coral are animals. So they have some qualities that make them look like rocks, and they have some qualities that make them behave like plants. But coral are really animals, and they reproduce usually once a year for a few days a year. And so they produce eggs and sperm. Actually, most coral are hermaphrodites. They produce both eggs and sperm. And but they have algae inside of them, and algae actually help feed them, so they have some plant-like qualities. And of course, they build these gorgeous reefs, and that gives them rock-like qualities. But really, it's better to think of coral more like building a skeleton. Much like the skeleton in your hand, we make it, but we're not rocks. We're living organisms. And the thin veneer of skin on my hand is similar to the type of thin tissue that's on the coral reefs. It's very thin, and people can damage it quite easily by walking on it. So when you're walking on the coral reef, you're actually killing a lot of those organisms.
So, and the coral reefs, of course, have tremendous sort of ecological importance as well as economic importance.
Correct. You know, when you think about the sort of ecosystem services that coral reefs give to us, it's tremendous. First of all, they're one of the oldest ecosystems on the planet, and they're just tremendously beautiful. I don't know a single person who has gone down to look at coral reefs and not come up going, wow. And so they're beautiful. They've been on our planet for over 200 million years, and a quarter of all marine life at some point lives on a coral reef. So they're very important as nurseries for small fish, but if you think of Nemo, which I often do, they're really the apartment houses of the ocean. They just house a number of organisms and protect them. And without them, those organisms leave. And so they provide homes, they're nurseries, but they also protect us. And they protect our cities and our homes from hurricanes and storms and tsunamis, for that matter, especially here in Hawaii. On the east side of our island of Oahu, we have very extensive coral reefs, and during the tsunamis, I was very glad that those reefs were there because my host was over on that side. They do provide a lot, plus they provide a number of pharmaceuticals and probably future pharmaceuticals for us, which are really important. And then on top of it, here in Hawaii and many places around the world, they provide a livelihood for us, for whether it's fishing or tourism, whatever it may be. And you spoke about the economic importance besides our future pharmaceuticals that we might get from coral reefs. Places like the Great Barrier Reef garner $6 billion, that with a B, not an M, $6 billion annually from the Great Barrier Reef. And I know that our reefs here in Hawaii contribute a great deal to our economy.
Yeah, they must work in a lot of different ways in terms of the tourism industry, the marine fishing, various types of things like that. And so the threats to coral come from a variety of different sources too, right?
Yes. So I try to break it down into local and global threats. And locally, there are many things that can contribute to sort of the stress on coral reefs. And I think Kaneohe Bay, I keep coming back to it, but it's a really good example of where we are in the world. Kaneohe Bay has had two major sort of local threats to it in the past, say, 100 years or so. And the first was the building of boat channels in Kaneohe Bay that dug up a lot of sediment and things like that blanketed the coral, that it certainly shifted the bay from more of a sandy bay to more of a muddy bay, even though there's beautiful sand there still. Also during the 60s and 70s, we had a sewage pipe that was in the bay that destroyed a lot of the coral in the bay. Some people say as much as 90 or more percent of the coral died during that time. But today, 35 years later, much of the coral that we see, especially in the northern part of the bay, is probably pre-World War II densities. I mean, in some of those places, you can't even stick your finger in on those little reefs, on those little patch reefs, because it's so dense with coral. So the coral have rebounded enormously well, but that doesn't, offset what's happening, globally. And globally, our oceans are warming all over the world, and that causes coral, who's lived in a sort of more or less uniform environment, say that plus or minus 8 degrees Celsius for thousands of years. And so when you boost them a little bit above their comfort zone, they stress. It's much like we do in some of our big cities during the summer. It may go up to 105 or 110 degrees. Older people and younger people stress, or people with with allergies or asthma, they stress and often have problems with that, and some may even die. That's the same with coral. But the other global thing that's happening is the oceans are becoming more acidic, or more like vinegar, and that is shifting the chemistry of the ocean, and that is due mostly to our overuse of fossil fuels and putting CO2 in our atmosphere. So it's very difficult to deal with those global causes. Every reef in the world is now subject to those global causes. And some of the work that we are doing here in Hawaii and at the Smithsonian University of Hawaii is one of the few things that can offset some of those global changes to our ocean, to our coral reefs.
Right, so that's, and I can see, because the oceans are so vast, so big, so deep, that as any change starts happening there, it has a sort of tremendous inertia. So I've heard it said, for instance, that even if we could stop atmospheric warming today, that the oceans will continue to warm for another 50 years, roughly, because of the inertia of the whole system, as it were.
Yeah, it's got a lot of heat capacity.
Right.
And so it's already absorbed a lot. And so, you know, as conservation biologists, we want to try and help organisms, whether they're in our zoos or whether they're in our oceans or, you know, in our forests, and help them survive as best we can. We clearly can't change right now the path that we're on. We could stop it, but as you said, that path will continue for some time. So our goal is to try and protect those organisms as much as we can.
Absolutely, absolutely. You have to do that. And the impacts, as you say, of not protecting the coral seem devastating. If, as you say, a quarter of the marine species use coral reefs at some point in their life system, if the coral reefs go, the whole marine ecosystem is vastly disrupted, presumably to ill effects.
Yeah, it's an experiment I don't think we really want to do. And the thing that's hard to know in this whole sort of changing world is that what the effects of, you know, the reefs becoming more threatened will have on us. Let's just do a thought experiment and say, okay, let's say 90% of all the reefs in the world were to go away. Certainly one of the things that would happen is that fish would become a luxury item. And so that affects us as humans, you know, not having the food that we like to eat and in some places need to eat. You know, in some of the smaller islands, or if you're in a sort of fishing community, you depend on that fish, that wild fish, for food. So it affects us personally, but it also has a larger and broader effect, because as coral reefs disappear, the fish often disappear. They may go other places, or they may just disappear. We don't honestly know the answer to that. But as these fish populations shift, the whole food web in the ocean shifts. I mean, it's not just coral reefs that are being affected. The food web in the ocean is now being impacted. Right. And this can affect whales, it can affect sharks, it can affect everything that's in the ocean. One of the things that we don't want it to affect is oxygen production. The oceans produce 50% of our oxygen on our planet. So it is so essential to life on Earth, and we just don't know how all these systems are intertwined, that we really want to make sure that we protect our basic and important ecosystems like coral reefs.
Wow, yeah. So what you're saying is that in essence we are right now conducting sort of global geoengineering experiments on a grand scale, sort of willy-nilly. And
well, I think there are very many smart people who are working on this, and I feel very optimistic, even though I know that people have predicted dire things. I think there are a lot of smart people who really can help with this issue, and so I think we will be good. But every person has to do their job, not just depend on scientists and engineers to get us out of this pickle.
Exactly. That's part of the reason we do this show, is to help promote the idea that science is something that everyone can do and everyone can contribute to, even without being a scientist yourself. So that's a good point. So give me a sense about what is happening in terms of how fast are coral reefs declining? Are they growing in some places, shrinking in others, staying the same? What's sort of the global balance?
Yeah, it's up and down in a variety of different places, and in Hawaii, I would say we're going like this. Some places it's shrinking, some places it's expanding. In other places like the Caribbean is perhaps the one ocean that's in the worst shape. Maybe the Indian Ocean would be following it, although the Indian Ocean is a much larger ocean. And I think the larger the ocean, the more the impacts are less, a little bit less. But each ocean is experiencing different things. But part of our problem is that because this is a, let's put this in quotes, relatively new issue, we have this problem of shifting baseline. And so I was really made aware of this personally when I go out into Kaneohe Bay, and I really enjoy the coral, and I go, wow, there's some really beautiful fish here, and I just go, wow, this is just great. And it is great. But when you see a picture of the northwestern Hawaiian Islands, the reefs in the northwestern Hawaiian Islands, and you see that the coral reefs are covered with fish, and there's sharks, and there's just so much teeming life, you understand that the main Hawaiian Islands are impacted, and that my perception is one of shifting baseline. And so it's a very difficult thing to deal with generation after generation, because my grandmother might have a very different view of Kaneohe Bay if she lived here, and what it looked like. But my view is that, oh, it's really good, but the fish really, you know, there's impacts in Kaneohe Bay, and clearly around the world as well. So this concept of shifting baseline is actually one of the more difficult problems we deal with in trying to get people to understand the depth and the breadth of this issue.
Well, that's intriguing. We're going to have to take a short break here. I'm Ethan Allen. I'm here with Dr. Mary Hagedorn from the Smithsonian. We're talking about coral conservation through cryopreservation.
Castle and Cook, Hawaii. Investing in Hawaii, creating communities, and providing for the needs of our state. Collateral Analytics, empowering the real estate industry to make better informed property investment decisions. The Foreign Trade Zone, bringing the benefits of the Foreign Trade Zone programs to Hawaii businesses and entrepreneurs. Galen Ho, a senior executive of BAE Systems, a global defense, security, and aerospace company. Hawaiian Electric Company and its affiliates Maui Electric and Hawaii Electric Light Company on Hawaii Island. The Hawaii Energy Policy Forum, incorporating diverse perspectives to design a flexible and forward-looking energy strategy. Hawaii Energy, the state's energy and efficiency program created to help Hawaii's residents and businesses adopt a clean energy lifestyle. Hawaii Gas, helping Hawaii achieve its transition to clean energy and a better energy future. Hawaii Pacific Health, bringing technology and teamwork together to transform healthcare in Hawaii. The High Tech Development Corporation, attached to DBED, is the state's leading technology agency. The Scheidler Family Foundation, supporting educational, cultural, and charitable organizations, including Think Tank.
And we're back. I'm Ethan Allen, and you're with us on Likeable Science on Think Tech Hawaii. I'm here with Dr. Mary Hagedorn from the Smithsonian's Conservation Biology Institute, and we're talking about coral conservation through cryopreservation. That's quite a mouthful. Basically, Mary does all kinds of interesting work with working to conserve coral by freezing them, freezing the sperm, the eggs, and the embryos, as I gather. And this is, I guess, the first question is sort of why do you do this freezing?
Let's step back to humans. Humans use in vitro fertilization and artificial insemination for a number of reasons, because they want to put off having a family for a while, or they may be having difficulty in terms of having a family. There's a whole host of reasons that people use this technology. And we are using this technology to try and preserve our genetic diversity on our reefs. Our reefs around the world are under threat, and I can't underline that enough, that our reefs are in trouble. And this technology is one of the few that we know of right now that can really offset some of the negative impacts of global climate change, warming and acidification of our oceans. And we started this work about 10 years ago at the Hawaii Institute of Marine Biology at the University of Hawaii, and just doing the basic science, because no one had actually ever applied this to coral. But it really is the same sorts of techniques. We create a coral fertility clinic that's exactly the same as any clinic you would find for humans. And we've been fortunate enough to be here on the reefs in Kaneohe to experience the coral spawns. And we are freezing the sperm and the embryonic cells of coral. And we've created a bank not only for some of the species here in Hawaii, but our largest, we have 9 species now around the world. We have some from the Caribbean, but our largest contribution right now is in Australia. And we've been going to the Great Barrier Reef with our collaborators there, the Australian Institute of Marine Science and Tauranga Zoo, for the last 3 years. And we have 6 reef building coral that are in banks there. And Tauranga Conservation Society Australia, actually that's its proper name, holds that bank for the country, obviously, of Australia. And we've also been using that bank. We don't just freeze things. We're now starting to use the bank. And again, just like you would in any fertility clinic, we brought sperm from the bank that had been there for about a year, and we created a large population of coral babies in Australia in December. And so we're starting to learn to use the bank so that we have a blueprint for the future. You know, our oceans may be quite different in the future, and we need to know how to best use the bank and rear the coral for the future. The material that's in the bank is good for hundreds of years, but perhaps even thousands of years. So people ask that question, like, what, you know, if you're going to just put it back into an ocean that's having trouble, what's the point of doing it? Well, there's a number of answers to that question. One is we can keep it until the oceans perhaps are more like they were 50 years ago, because the cells are frozen but alive, just like in a human bank. And the other thing is that we're using this bank to potentially allow for selective breeding. And I have colleagues now that are interested in trying to build more resilient populations, ones that are more robust to temperature changes and changes in acidity. And so this bank will play a huge role in the future of those experiments.
Excellent. Well, that's very exciting. So you say you've got 9 different species of coral here and 6 down in Australia. Do you have to treat them differently, or do you pretty much apply the same sort of process to each? Do you tweak it slightly? How does that go?
There's 9 total. We use more or less the same process for all the coral sperm. We have a slightly different process for the coral embryonic cells, but what we try to endeavor to do is make it very field friendly. And we keep, even though we have very expensive equipment in the laboratory to do this, we've created very inexpensive methods and ways of freezing that any person in any country can afford to do, and we can train them to do it very quickly. So the idea was to really get this to work, and then to try and start training people around the world, which we're still not doing. We don't have the funds and resources yet to do that, but it's high on our list to do. But we have very simple methods to do it now, and we believe that in a few years many countries will be doing this and making their own banks.
Well, that's great. So you're trying to sort of get your work to be replicated and to become sort of self-sustaining in that way.
Correct.
Excellent, excellent. Because of course, different reefs around the world have different sets and different subpopulations of the same species that may have different characteristics.
Exactly.
Plus, there are many, many, many species of coral, right? I mean,
there's about 800 species of stony coral, okay. And those are the ones that are perhaps the most at risk at this particular moment in time. And there's, as you know, as you were mentioning, there's a lot of genetic diversity that we still have in our oceans. But the thing that can happen quite quickly, and I didn't mention this previously, is that there's a lot of stress on our reefs, both from global and local causes. And this can cause local diseases to get a foothold quite quickly. And so, you know, you can see a disease go through a coral population within weeks, or a bleaching event can wipe out, you know, reefs in a heartbeat. You can lose diversity so quickly. So by allowing these techniques to be all over the world, I'm hoping within the next 10 years that the majority of our coral species will be banked in some way. Now, you know, you think, oh my gosh, that's such a large task. you really only need 35 individuals to be banked for each species, which is a very, very doable task and approachable, you know, especially if you had key people in different countries doing, say, 5 or 10 species a year. You would quickly have much of the world's coral population in a repository.
Excellent. And it also sounds like then that requires good collaboration and communication among the scientists doing this work. So you know, as you say, you're working on the right species in the right places at the right times.
Yes, and you know, I think in general the coral community is very very knowledgeable and knows a great deal about coral reproduction, even though it's only happens a few times a year. It was so impressive with the Great Barrier Reef, you know, the massive spawn on the Great Barrier Reef, that, you know, a lot of scientists quickly got into it and love coral, obviously, as I do. So, you know, I think the idea is that we would form a team, a network of scientists that would help each other, you know, technologically, even physically if they needed it. But the idea is to have kind of a SWAT team for this really important threat to our oceans, and to work on it quickly meaning the next 10 years. I guess that's not so quick, but for scientists that's pretty quick.
No, that's a nice metaphor in a way, that the network of scientists collaborating as the coral animals collaborate to build the reefs.
Exactly.
In a sense, yes. And there must, in addition though to this, to your cryopreservation stuff, there are other efforts going on to preserve coral, I assume.
Oh yes, and you know, I think I need to underline too that every conservation effort that we are using to try and save coral reefs are important. And cryopreservation is just one of the tools. There are people who are trying to create marine protected parks. There are people who are making coral gardens, and they fragment the coral and they plant them in a variety of different ways. I mean, there are many different things that are going on in terms of conservation, and all of them very important.
Excellent. Well, that I guess is one of the reasons why you're hopeful that the situation may resolve positively. Certainly, that's got to be high on the humanity's priority list, I think.
One would hope.
And so you talked about your work here. You mentioned your work in Australia. You briefly mentioned the Caribbean that you've worked in, and indicated they've got real, even perhaps deeper challenges.
Yes.
Are they also banking there?
Well, we did bank. There are 2 species on the endangered species list in the Caribbean. At one time, you know, so you need to think of a coral reef as a forest. You have tall trees, and then you have small shrubs and vines and things like that. And all the species kind of fit in. So the tall trees are what we call the reef building corals. And those are some of the more important ones to create the reef, or create the forest, if you want to use that analogy. And in the Caribbean, you can still see these species that are on the endangered species list, but they are no longer a functional ecosystem. And they're scattered, and in some cases, we're not even sure that they're reproducing reliably, which is really bad. So we have cryopreserved some of the 2 endangered species, but certainly not enough. When we were there in Belize is one of the places, it's the oldest coral reef in the Americas, the second largest after the Great Barrier Reef. And it seems like age helps with some of the coral reefs in terms of their longevity of reproduction. And when we were there, we were just about to do all this cryopreservation, and we had to evacuate because 2 hurricanes were barreling at us. So we quickly grabbed our material, and we had frozen thermoses that we took with us, but we only got a portion done of what we would have liked to have done.
Oh, that's too bad, because it sounds like a critical effort there.
Yes, and I would like to expand in the Caribbean. We just right now don't have the resources to do it.
And then the Indian Ocean you mentioned also.
I have not done any work in the Indian Ocean, and I would love at some point the invitation to both train and do some work in the Indian Ocean to get that started.
That would be great. Are there folks over there doing anything like this?
Currently, our laboratory is the only laboratory in the world doing this work. We have collaborators that we work with, but generally we're about the only group doing it.
it seems like you very much need to need to get that expansion moving.
Exactly.
Excellent. That'll be an exciting next challenge for you,
Yes.
I'm sure. I think we'll take another break here. Again, you're with us on Likeable Science. I'm Ethan Allen. I'm here with Dr. Mary Hagedorn from the Smithsonian's Conservation Biology Institute, and we'll be right back.
Aloha, I'm Nicole Horry for ThinkTech. For nearly half a century, the Hawaii Foreign Trade Zone No. 9 has brought the benefits of the Foreign Trade Zone program to Hawaii businesses and entrepreneurs. DBET, the Department of Business, Economic Development, and Tourism, operates Hawaii's Foreign Trade Zone program to encourage international business and economic development. The Foreign Trade Zone's mission is to increase the amount of international trading activity in Hawaii, thereby providing employment opportunities for the residents of our island state. For more information, see ftz9.org. I'm Nicole Horry. Mahalo.
And we're back. I'm Ethan Allen. You're here on Likeable Science on ThinkTech Hawaii, broadcasting from the studios in Pioneer Plaza. With me today in the studio is Dr. Mary Hagedorn from the Smithsonian's Conservation Biology Institute, and we're talking about coral conservation through cryopreservation. So, you just mentioned in our previous section there about being driven out of the Caribbean by a hurricane in the middle of one of your coral gathering projects, and given the very limited sort of time that you have, because the coral all spawn synchronously, that seems like that must be a huge sort of technical challenge. And you hinted earlier that the way of freezing sperm or eggs or embryos of coral is somewhat different too. So it sounds, can you expand a little bit on these sorts of challenges that you're facing?
Sure. So one of the biggest challenges that we have, aside from people and resources, obviously, is hitting the timing right. Generally, I can tell you in Kaneohe Bay, I can look at my watch and say, I know exactly when the next coral spawn is going to be. But in some of the other oceans, especially the Caribbean, it's shifting. It's starting to shift. So one year we had a huge expedition all lined up. Probably 20 people were coming with us. And I got a call from the folks who were already down there in July. They were supposed to spawn in August. They said the coral spawned. We missed it. We completely missed it, and we had to wait a year. And so it's a complete waste of resources and people and money. And so getting to some of these really remote spots to hit the timing right is really important. We have a challenge coming up in this winter when we go to the Great Barrier Reef. We have a thing called a split spawn. And what that means is that there's 2 full moons in December and November that could potentially stimulate the coral to spawn. Now, coral are animals, we're going to review that, but they respond to the sun and the moon and the stars. And so they can track the seasons and the sun and the warming oceans, and they spawn synchronously in response to that. But sometimes the cues are very close together, and it's called a split spawn. And you don't know which month to go in. Is it November? Is it December? Sometimes you make the wrong decision, and certainly, when we were working in Puerto Rico, we chose the wrong month. And so you're sitting there just like the coral are not spawning. So it's the biology is very challenging, and that's why for me it's been a pleasure to work in Hawaii, because unlike other places in the world, the coral in Hawaii spawn throughout the summer. So I'll start April 16th at 7 a.m. and go until September. And different species, we have about 4 or 5 species that spawn in Kaneohe Bay, and some are in April, some are in June, some are in August, some are in September. So you can have a coral spawning every month in Hawaii. So it's a great place to work on coral reproduction.
Excellent. So what you're saying though is that really the sort of the precise factors that stimulate coral to spawn are not thoroughly understood, in the sense that you cannot well, outside of places like Kaneohe Bay, you cannot reliably predict sometimes when your coral are going to spawn.
I think they are well known, quite frankly, but I think conditions are starting to change. And so, for instance, in the Caribbean, when we missed the whole spawn by a month, the ocean was really warm. And so instead of spawning in August, they spawned in July because the temperature got to the right point, and they said, now's the time. But it, you know, perhaps if you had looked 30 years earlier, that never would have happened. You know, we just don't know. We don't know enough about coral reproduction. It's only been known since the mid-'80s, and so our understanding of how it used to be and how it is now is very short. And now that we're changing our oceans, it's just very challenging to know what's happening because of the oceans are changing or just because of the variability in coral. So
yeah, so this is then very parallel to things we actually see in terrestrial environments too, where in some of the islands here, at certain months of the year when breadfruit were supposed to always be ripe, and there are cultural ceremonies involving breadfruit and all. Now they can't predict that anymore. The breadfruit may be ripe months earlier or months later, and it's being culturally disruptive because the plants themselves are now off of their traditional cycle. So
exactly.
I see. So this is not just that they don't know about this, but you're facing a very changing baseline, a shifting baseline kind of situation.
Yes.
Excellent. Ooh. Frightening level of challenge. So you mentioned the other thing was sort of personnel and funding. Those I know in all scientific research are chronic challenges. Can you tell us a little more about what you're trying to do to face on those?
So some of the challenges are actually in terms of basic science. Cryobiology is not a common subject, even taught in basic biology. So it's the biophysics of cells at cold temperatures. And even though you might take a class in basic biology or in membrane biology or whatever, it's very rare that you would learn anything about cryobiology unless you're going to go in and become an in vitro specialist. And so getting trained personnel is one of the challenges. And so it's very difficult to get graduate students and postdocs who are knowledgeable coming in. The other is, really getting the money to both support them and to get out into the field to some of these very remote sites where we need to go. A field season can cost, for say a group of 6, can cost about $20,000, and that's not a lot of money to bank down a species or even 2. But it's challenging for us to come up with, and it's the kind of work we do falls in between the National Science Foundation, which does basic science, and non-governmental organizations like say World Wildlife or something like that, that do more in situ, which means in place kinds of conservation. So we do a thing called ex situ, which is out of place. We take the organisms and we conserve them away from their place of origin. And this type of ex situ conservation is not, it just falls in a notch. And so it's often very challenging to find even foundations and groups that are willing to say, yes, this is a good approach. We like this approach. And so on all levels, it's really been quite challenging.
So any rich donors out there? I know Mary would love to have you get behind her cause.
Exactly. Send bags of money. We're open.
No, but you bring up a very good point that science proceeds along sort of fairly traditional routes, and you're sort of carving a new path, and that means you don't fall within anyone's comfort zone, sort of.
Exactly.
And so the funding agencies, which tend to be fairly conservative about how they award money, become hesitant about funding you. Yeah.
Exactly. And I think people don't understand too that science, like any other endeavor in humans, has fads and fashions, and so something that might be very popular in 1980 is no longer popular now, and I think you just have to be cognizant that things do change, and getting the word out is really one of the most important things, because once people say, yes, this is really important, we really want you to do this, it trickles up or down or over to these funding agencies. And so I'm very delighted to be here with you today.
Well, no, and again, this is beautiful, because you point out the value of this bank for preserving the diversity of genetic lines and different forms of coral across the whole surface of the globe, and at the same time we've talked about the absolute need for coral reefs in terms of what happens if they start deteriorating in terms of the human costs, economic costs, ecological costs, the whole ocean food chain basically falling apart.
Well, I don't think it's that. I think it's important, and I think that the key element is not only is it for the future, it's for today. And so we are trying to create new populations, and so that as populations begin to dwindle and shrink, we can offer a service and say, we can help you. We can try to diversify your population, or we can try and restore. If there's been a grounding or some problem with the coral reef, we could help.
That's excellent. That that kind of stuff is so critical in the world today. So your work spans this wonderful range from this very basic research up to this very practical, hands-on, applied science. So that's an amazing range of work.
Thank you.
You were to be praised and congratulated for that.
Thank you. Just bags of money Ethan, would be good.
Do you hear that out there? all science, I know all science needs money, and we all know funds are tight, but that's critically important, and certainly the education components of this too, as you point out, to get the trained people involved, to get the people with the right kinds of background, particularly I'd imagine some of these more remote sites, to get the populations to understand that their coral is in danger, what happens if it disappears, how they can themselves help protect it. All these are...
You bring up a good point. Training on many levels is something that we're interested, not only to train people to do this cryopreservation, but also we're interested in training people in basic science. We have a new project that we may talk a little bit more about, which is called Marine Geo Hawaii, which is just gearing up now. And one of the goals of that is to train people in Hawaii and in the Pacific in basic marine sciences, and also to have citizen scientists and high school students participate, whether they're here in Hawaii or in the outer islands, and to get interested and engaged in our oceans. And I think it'll be a tremendous success, and we hope it will be a long-term monitoring. It will be a long-term monitoring project for the next 30 years. We hope it will be very successful in spreading the word and the education and knowledge about our oceans to the Pacific.
That sounds certainly absolutely vital, and it seems like it fits in with other ongoing efforts for addressing climate change education that I know are ongoing out here through a variety of different organizations, and sustainability programs in a lot of different parts of the environment. So this Marine Geo sounds like a really great endeavor. I hope I hope to see more from it. What's the status of it as a project at this point? So
it's a Smithsonian-led project, but it is a collaborative project at heart. It mimics or parallels a similar project called Forest Geo in the Smithsonian, and that started about 35 years ago. And the question they asked was very simple, and it was, why are tropical forests more diverse than temperate forests? And they took a plot, a 50-hectare plot, and they measured every tree, and they got the species of every tree in that plot. Now, 30 years later, there's about 45 to 70 institutions that are partnered with the Smithsonian on this. They all do their plots exactly the same way, but they've added a great deal of complexity to those plots as new partners come on and want to do mammals or bacteria or whatever it may be. And so today now, we have a snapshot of forests around the world in different longitude and latitudes, and it gives us a really good idea of what's happening to the forests as our atmosphere changes. We hope to do exactly the same thing in the oceans and help our partner sites and lead them, and so I think it will give us a lot of really good information about how our oceans are changing over the next 30 years.
Wow, that sounds like very exciting work, because then it will build on that, and yes, you'll have the replication of data that is so central to science. You'll have the multiple observations of multiple sites of the same and different species of coral under similar and different kinds of conditions with similar and different kinds of threats, and can really hopefully then draw very, very good conclusions, figure out the solutions to the problems, see if replacing one species with another is a viable option, or helping to crossbreed species or some such may be yet the way to go. Huh, fascinating, fascinating. How do folks get involved in this Marine Geo project?
Well, we just had a recent workshop last week to invite partners. We're always open to more partners, and they would email me, and we would just bring them into. We want it to be both a scientific endeavor, but also one that involves the community and is sensitive to our culture here in Hawaii. So we hope it will be very inclusive and have participants from a variety of different education levels, from K through 12 through citizen scientists to graduate students and undergraduates. We're hoping that all of these groups ultimately will participate in this at some point.
Well, that sounds like a very ambitious undertaking.
More bags of money, I think.
But it appears you've been able to approach some of these very challenging tasks and succeed at them already, so I have no doubt that you'll thrive in this one here. Let's take a little bit of a look towards the future, if we might. So where do you see your bank going? What's going to happen in the next 10 years?
It's a really good question. Well, hopefully what we'll have is a distributed bank around the world. So there's a bank already in Australia. We have a bank. Actually, the coral is sitting. The U.S. coral is sitting in a bank in at Fort Collins. The U.S. Department of Agriculture hosts our bank for us, and I'd very much like to have a bank here in Hawaii. I don't have one, and I'd like the Hawaiian species to be in the Hawaiian bank. You know, I would like every country to have a bank and use it as they see fit. And that they share that material, so we have parallel structure. So if something were to happen, say to Mexico, for instance, not that we want something to happen, but say a hurricane hit the bank in Mexico, we can then say, here's more material back, you know, a growing collaboration and partnership, I think, amongst nations that are interested in securing their genetic diversity of their reefs.
Wow, that'd be great. That's sort of, you paint a wonderful picture there.
Yeah, a little Pollyanna.
Oh, well, you need to think big, to you know, dream large, and
Exactly.
and push ahead, as it seems like you're certainly doing. So that's quite admirable indeed. So is that how you will sort of know you've succeeded? Is when you begin to see more people doing your work, more banks popping up, more people applying your techniques in more places?
I think that's right, Ethan. You know, I mean, it's great to be a pioneer, but it's a double-edged sword. You know, you maybe you're the only person doing it, and that's really great at some point. But you also don't get the funds. You don't get the people who are who can be collaborators and teammates. And so having more people doing this type of work and conserving our genetic diversity, that's at the heart of everything. And allowing us to go a little bit faster, that's when I'll feel like I'm truly successful with this project.
Excellent. Well, that's certainly a grand goal to shoot for, and we really need to do that. Let me shift, if I might, for a moment and talk a little more broadly about this conservation issue. The Smithsonian has been involved, I know, in conservation for some while. Can you talk about some of their other efforts a little bit and give us, maybe put yours in sort of a context there?
Sure. So as you so kindly said, I'm from the Smithsonian Conservation Biology Institute, and it has been involved in ex situ conservation for about 35 years. And some of the projects have ranged from helping to save the Florida panther to the black-footed ferret. It is one of the first programs that actually showed that you could cryopreserve sperm from a very small group of black-footed ferrets that were thought to be endangered. 20 were found on the prairie. There are now 4,000 that are on the northern plains, and it's been a very successful endeavor. To the giant panda. The Smithsonian has been partnering with conservation biologists and veterinarians in China to help them improve the health and the reproductive capacity of the giant panda. And we have participated in creating banks for a number of large cats, clouded leopards, cheetahs, etc. So there's been a lot of ex situ conservation done in my immediate group, But also, on the larger scale of things, there's a number of large terrestrial forest plots and things like that. I mentioned forest geo, but there's a large number of terrestrial things on elephants and the forests in Asia. I mean, we are developing into one of the premier conservation organizations in the world because of this breadth of both in situ and ex situ approaches.
Super, super. We're going to take another brief break here. I'm Ethan. Oh, sorry. I guess we're actually going to wrap it on up here. So you've been listening to Likeable Science here on Think Tech Hawaii with I'm your host, Ethan Allen, and with me today has been Dr. Mary Hagedorn from the Smithsonian Institution's Conservation Biology Group, and we've been talking about coral conservation through cryopreservation. I very much appreciate all your great insights here, Mary, and you've given us a wonderful overview of the whole situation. The challenges you face are immense, but your passion is tells me you'll meet these challenges.
Thank you, Ethan. It's been a pleasure.
Well, thank you so much. Good.